Good afternoon. First, I would like to thank Dr. Nagla Abdallah for inviting me to share with you some of our teamwork. We will talk about development of transgenic wheat with improved biotic and abiotic stress tolerance or environmental stress tolerance. First, as you all know, this is the types of environmental stress, biotic stresses or abiotic stresses. I will focus today to talk about, for biotic stress, about fungi stress, and in abiotic stress, we will talk about drought stress and salinity stress. There are different approaches that scientists adapted to improve stress tolerance using genetic engineering. This includes the production of osmoprotective compound in the plant, like what we will talk about mannitol production, or engineer the plants with molecules that protect macromolecules like late embryogenesis abundant protein, or some other things like regulatory proteins or ROS scavengers. So when we first started this work, maybe around 10 years ago, the first thing we think about was to establish a regeneration and transformation system for hexaploid, especially for Egyptian cultivars. This is because in wheat, especially, the transformation and regeneration is genotype dependent. So our protocols depend on we use the immature embryo as the explant, okay? And we induce the culli for 5 days, then bombard the culli with whatever vector we want to introduce, and then we let the culli on non-selective media for 1 week, and then we transfer to selective media for other 3 weeks. And then transfer to regeneration media and rotting media, and this is acclimatization where we leaf bend the plants to distinguish between the transgenic and non-transgenic plants using basta herbicide. Of course, in all of our transgenic, we introduce bar gene, which will have the effect or no effect for transgenic for this herbicide, or it will be herbicide resistance of the plant are transgenic. And after that, the protocol, because we are introducing the bar gene, we can select the segregating lines reaching to the homozygous line before we do any field testing using the bar gene expression. So just spray the basta. This is non-transgenic plants, just will go die. And in here, you can just count the segregating families to know even this give you an idea about the copy number of the introduced gene. This is more close photo for another family. And in here, this is distinguished between transgenic and non-transgenic plant. Okay, what the genes that we are adapted to use it in our lab for abiotic stress, HbA1 and MTLD, this I will talk about them today, and how to like, I think, Shirin Abul Maati, the student here at Cairo University, her PhD work about this gene. And for biotic stress, we are adapted to genes. The barley chitinase gene, this is for fungal resistance, and avidin, this is for insect resistance, but I will talk today about only the barley chitinase. Okay, now we'll talk about HbA1 gene for drought tolerance. HbA1 gene is one of late embryogenesis abundant protein. So it will protect the macromolecule when the plant have stress. Of course, there is another experiments that use model plants like tobacco using this gene before. We subclone this gene into a plant expression vector according with the power gene, which is our selectable marker. And we, of course, will do all the molecular analysis, but I will share with you the Western blot analysis. In here, this is a HB1 gene. It's already expressed in wheat, okay? But when it expressed only at the seeds, because it's late embryogenic abundant protein. If you take a sample, you cannot, from the leaves or from the roots, you will not find any match. So this is the seeds of the plant, or all of them, even transgenic or non-transgenic, is expressing the gene. But when you take the sample from the leaves, you found only the expression in the transgenic plant. So by this, we improve that we insert the gene and also the gene is expressed. After that, we make like a very small experiment at the field of our institute to compare between the control, which have received 8 irrigation per season, and with drought, which just received 2 irrigations. And we find no significant difference in between. So, of course, for the transgenic plant comparing to non-transgenic wild type. So this encourages us to take this to the north coast of Egypt to make like an experiment at area called Rafah. I think you all know, we cannot go now there. And this is rain-fed experiment. Okay, and after that, we, the year after, we take it to all the long coast of Egypt from Marsa Matruh to Sinai. Okay, and we found by using also, it is a replicated experiment, and by statistical analysis, we found that the transgenic gain more 20% of the grain yield when comparing to its wild type non-transgenic plant. And we published this 2005. Now I will talk about another gene. It's for salt tolerance. It's MTLD gene. It's a mannitol accumulating gene. This gene is isolated from E. coli, and it encodes for enzyme called mannitol phosphate dehydrogenase. It's thought to be an osmoprotectant or one of something called compatible solutes. Some reviews said it's also work as ROS scavengers. So also, using the same strategy, we subclone the MTLD gene under constitutive promoter, which is ubiquitin promoter, along with our selectable marker bar gene. And this is some molecular analysis. This is in here, this is southern genome reconstruction. We wanted to detect the copy, the transgenic copy number by doing this, and we have 2 transgenics, which we name it 79 and 235, which maybe have 1 copy of each. And in here we do also the BAT assay. Okay, this is for bar gene expression to prove also that we have a transgenic. And this is the Northern analysis to prove that the expression of the transgene. After that, now we are doing some analysis because we are introducing sugar alcohol. Okay, we want to investigate whether this sugar alcohol interrupts other sugars in the transgenic plant. So in here, you can find this is the total soluble sugar. This is the transgenic. You can find in the grains total soluble sugar are increased. In the leaves, it's increased significantly. If you compare this with the total sugar in the grains, the difference may be going to non-transgenic line, while in here, it's also all in the plant, it's also the same. So for total sugar, to compare this, you can find that the soluble sugar or some of non-soluble sugar are converted to soluble sugar. Of course, we make this is led to we wanted to investigate which sugars that was interrupted by our transgene. Of course, the mannitol, you cannot find the mannitol in our wild type plants because wheat is not mannitol producing. Okay, but you can find it in only in our transgenic. So there is no bar in here. Also, we found in the fructose it is elevated in both transgenic lines in here, in both grains and plantlets, and the same for the glucose. So the sugar profile, all of it are changed. And we are now preparing to predict the pathway which led to this interruption in the content of sugars. Also make like pilot experiment to see if this transgenic or this gene has an effect for salt tolerance. You can find in here also, of course, we take all the parameters for yield and yield-related relates and sand culture experiment. We can find in here, unless the vegetative growth it's not affected or it's not increased in transgenic plant, we can find that the grain yield are increased in the transgenic comparing to the non-transgenic plant. And this is reflecting in the reduction percentage under the stress. So the gene gives some tolerance to the plant, but now what we are investigating right now, is this because of mannitol or is this because of the change in other sugars in the plant? Now the other topic, which is production of bread wheat resistant to rust and powdery mildew. As you know, the rust diseases, we have stem rust, leaf rust, and yellow rust, and also, I think all of you know about powdery mildew. The strategy we adopted, the rust or the fungal that cause the Buxenia fungal have a chitin in its cell wall. Okay? There is many reports said that when you use a gene that expressed chitinase, this chitinase gene may lead to the non-growth of the fungal. Okay? So we take the chitinase gene. Actually, this gene was isolated in our institute by another team, and we take it also to put it under ubiquitin promoter with our selectable marker par gene. We got 72 different transgenic lines, and we propagate. This is happened in 2005, season 2005-2006. And we take those putative transgenic and we plant each one in single row to get the homozygous line. Of course, this is the first thing we did in molecular analysis, which is a PCR. And in here, there is a problem about chitinase gene. Also, because the chitinase gene is, there is an indigenous chitinase in the wheat. And our gene is already isolated from barley, which are very close to the sequence of the barley chitinase. So to investigate if the gene is there, you cannot use just specific primers for the our introduced gene. So we only get to use the primers of NOS terminator with the forward primers of chitinase, because all the results will give us positive in the first try. And this is shows certain analysis for 2 of the transgenic, not all of them, of course. We have all the figures, other figures. And this is an segregating, this when we plant our transgenic in the field of Agiri to see where is the homozygous lines or homozygous families and the segregating families. And you can see here, this family is still segregated because some lines are died, while this family and this family reaches the homozygosity. And this is more close figure. Okay, and this is in another place in Gemese Station. This is a homozygous family, while those are still segregating families. So we goes to real-time PCR to compare the expression between the different lines, to choose the best of them to go for field testing. You can find here this line is more expressing, and then of course the high line, the chitinase gene. And in here, this is the chitinase activity analysis. We will find the same lines which have chitinase activity, comparing which differ significantly from the non-transgenic line. We went to field evaluation, and this is the spreaders. We plant plants, and this is the borders which we infested with the rust and powdery mildew, and this is the shape of the infection around our plants. And in here you can compare. This is non-transgenic and transgenic. Some of our transgenic even didn't get one spot of the fungus, and some have like a moderate infection. This is of course according to the transgen expression, or how it is located, and this data was correlated with the chitinase activity data and with the real-time PCR data. And this is some of the transgenic. Okay, this was surrounded by the pictures that you saw before. And this is some picture of field visit 2009-2011. You maybe see some faces in here. Okay, this is Dr. Taimur. This is Dr. Mustafa Shami, which is who is working in Plant Pathogen Research Institute. Okay. This is Dr. Madkour, Dr. Bahai, who was our team leader. Okay, and we are now, we got the homozygous family, and now we are propagating this family. Actually, we stopped for 1 year because what is happening last year, so our data stopped at 2011. Okay, you can find in here, this is non-expressing transgenic line. It takes ATS for leaf rust like the non-transgenic, and this is the reaction of our old transgenics. And we investigate if this affects the grain weight or not. You cannot find, there is no significant difference in between. So now, because we are working with GM crops, and we went to field testing, we have to satisfy the requirements of National Biosafety Committee. So all this work have done under the investigation of the National Biosafety Committee, and we make some risk assessment during these experiments, and we did some compositional analysis for our grains. So first, also we prepared the dossiers that have all the information required according to the EFSA instruction. And we did, this is only for HbA1 transgenic. We made all the compositional analysis according to the consensus document for wheat. And this is data. I think our transgenic in the
systems, and that is what we moved forward to carry out further studies. And what we did then, after number one is the species selection, so we had decided on the 10 based on their abundance. We now cultivated them in the lab to grow enough for lipid extraction. And then after we did that, we chose 2 species, as I will mention in a few minutes, to try and see if we can reproduce them en masse in actual ponds. In other words, slowly driving towards our main aim of going to the rural or to the communities, because it would defeat the purpose if we could not move from the lab to the communities. So this was the kind of setup we had. We had to isolate each of those and have monocultures and grow them, like I said, in enough quantities. And then after that, we did lipid extraction, and we were able to see which species had the most lipids in them and therefore had the potential for high yield of the biofuel eventually. And you can see from this graph that we have Chlorella species was the highest, followed by Euglena, and then we had Nischia species. So from there, we now zeroed in to these three, and we tried to characterize, just chemical characterization of the algae to find out what kind of glycerides they had, to begin to have a better understanding of what oils they have the potential to produce. Now, looking at this table, I would like you to just really pay attention to this. This is from Euglena species. What is important about this, apart from being high, is that it's a unsaturated glyceride, and what that says is that it has high oxidative stability, and therefore if you were to use that oil for further processing, you had a higher chance of success than if you used the poly unsaturated speed. That's all I wanted to point out. So we sort of zeroed in on Euglena and Chlorella because they had the highest amounts of the glycerides. Now this is an example of the pond that we used. We actually went to an institute that already had ponds where they grow fish, and we were able to get 2 of these ponds and to grow these 2 species, Chlorella and Euglena, to see what kind of production we could get out there in the open, because this will then mimic what we are likely to have in a rural community. And we were able to reach, after about 20 days of culturing, we were able to reach 2.4 grams per square meter per day within these ponds. This is the example for Euglena. And therefore, in concluding, we realized that actually we have algae that is widespread in our lakes, and therefore we had natural sources that we could use. We also concluded that lipids were observed in dominant species and in amounts that were commercially viable, and we identified the species that had these high yields. And then we also noted that the algae oil, we had both polyunsaturated and monounsaturated fatty acids as making up the glycerides in these oils, and that microalgae can therefore be a potential source of biofuel for Kenya. This is sort of preliminary, but we're still going on with the project, and the next phase is actually to go into mass production and hopefully link up. We already have linked up with an industrial person who will take this up on an industrial scale and get it rolling down. Now, this work is still ongoing, and it was supported by a grant from the Kenya National Council for Science and Technology. Thank you, Mr. Chairman.